event in more than a year, which is really a remarkable thing. Which brings us to tonight. This is a great conversation that I'm so looking forward to. Our special guest tonight is Lance Butler, and I've been wanting to do this because I know so little about freshwater mussels, and I know that they're so important. In fact, they're the most imperiled animals in both America and the Delaware River watershed, and restoration of mussels has become a national priority, especially as these hard-working animals perform a vital service of filtering our water. A coalition of public and private partners has ambitious plans, including propagating as many as 1 million mussels annually in labs and ponds to restore their populations. So although I know a lot about birds and wildflowers and insects, I've been teaching for 40 years, I know so little about mollusks, and that's why I'm looking forward to tonight. I'm finally going to get to learn something. Our guest is Lance Butler, a friend and senior scientist with the Philadelphia Water Department's Office of Watersheds. He's been with the city for 22 years, where his focus remains on restoration and functional ecology and urban ecosystems. Lance holds a Master of Science in Aquatic Biology and is pursuing his PhD with Drexel. His research aims to identify the benefits of freshwater mussel restoration in urban systems, and we'll be exploring the whys of that tonight. He's also been involved in the planning, designing, and monitoring of programs associated with the Flat Rock Dam Manayunk Canal Improvements Project. He's our Roxborough neighbor, neighbor of the Schuylkill Center, so we're happy to claim him as one of our own. Welcome, Lance.
Thank you. Thank you very much.
So glad you're here.
I'm glad to be here.
We're going to let you talk for a while, and then
Sure.
Everybody listening in, if you have questions as Lance talks, put them in chat. And when Lance is done, I'll start reading your chat questions to him. Thank you, Lance. Welcome.
Okay. Bear with me for a second, and I'll start this up. I'm hoping everybody can see this. Good evening, and thank you for having me to present on the blue collar bivalve. Basically, what I want to talk to you about today is how we, the Water Department and other agencies and other groups within the city of Philadelphia and the regions of the Delaware Basin are investing in this unique organism, something that's been overlooked for years, thousands of years in many cases. Was once a novelty in our Native American heritage, but has since gone by the wayside due to its lack of commercialism. It's not an edible mussel, as you would think. Most people think freshwater mussels or they think of oysters, and this is not the case. So I wanted to talk to you about who's involved first. First, in 2018, we, Philadelphia Water Department, signed a memorandum of understanding. We created this concept called the Aquatic Research and Restoration Group. And it was consisted of Philadelphia Water Department, Academy of Natural Sciences, the Partnership for Delaware Estuary, Bartram's Garden, Independent Seaport Museum, and the Philadelphia Parks and Recreation. In addition to that, we've since gained another incredible institution, the Audubon, Pennsylvania, which I think, I don't know, you've all been to their Discovery Center yet, but is absolutely fabulous. So I hope you do attend there. And so the initial charge of the ARC, what we call it, the Aquatic Research and Restoration Center, was to develop a freshwater mussel hatchery within the confines of the Fairmount Waterworks Interpretive Center. And this hatchery was supposed to be, or is, a public education outreach type of laboratory, but it's also a breathing, living laboratory. So we're actually doing research and development here. And in doing so, we developed a technical advisory group from PWD, PDE, and Academy of Natural Sciences. So you would ask, why is the Water Department of all interested in freshwater mussels? That's a great question. And the reason is there's 5 major reasons that we are interested in these unique organisms. First of all, foremost, is that water quality benefits. These organisms can filter a incredible amount of water in a given day. So a mussel in the Delaware River right now one mussel can filter 10 gallons per day. So if you think about 1 million mussels, you're talking about 10 million gallons and keep on going with that exponentially. Again, their ability also to biofiltrate or to remove pollution and sequester pollution. utilize some of the pollutants like nitrogen, phosphorus for shell growth. So that's biosequestration. They're excellent bioindicators too. These organisms are long-lived. Most people think, mussel 3, 4, 5 years old. No, actually, they're 30 to 100 years old. They can live these long life cycles like ours. And they are very, very sensitive to certain anthropogenic or human-induced type of pollutants, like human waste, ammonia, also heavy metals. These things they're very sensitive to them. So they're great canaries in the coal mine for our waterways. Extremely complex life cycle. Most people think, when they think of mussels, they think the spat, like in oysters, are broadcast and they attach to a shell in the bottom and live out their life. Well, in reality, a mussel, freshwater mussel, can't live without fish. And it's basically an evolutionary response. A mussel can't move or can't move great distances. So if you think about a fish, it's their Uber to get upstream or downstream. So what happens in their life cycle is that the reproduction usually occurs in the fall and the winter. And then the female actually broods the babies in her mantle or in her region with the gills in these huge tubes. And these things are called Glaucidia, and they're larval stages. And then during the spring, when their host fish come by, they disperse, or they have 3 different types of mechanisms how they disperse their Glaucidia or larvae. And in one way, they basically mass disperse, and the fish breathes them in, and they attach onto the gills of the fish. And again, they hook onto that ride for anywhere from 10 to 15 to 30 days, depending on the species. So it's very species specific. They're also very sedentary type of organisms. So their life cycle might be in a 10 square meter area. And in doing so, they can't run away from pollution. So again, great canaries in the coal mine. The other unique thing is that they're ecosystem engineers, and that word or concept has been tossed around a lot. And it's basically, they have the ability to terraform. Because they're filtering so much water, they can clear the water. In doing so, light can penetrate and vegetation can grow. At the same time, they create a very rough bottom with their umbos or their beaks sticking out, so the roughness is increased. And they create interstitial areas for macroinvertebrates and small juveniles and fish and so forth. So they create a 3-dimensional canopy or habitat. And like Michael said, they are the most imperiled species in the United States. Out of the 270 plus species, 70% are either in federally or state listed and endangered. So the goals of the aquatic research and restoration were to obviously successfully propagate these freshwater mussels in laboratory, multiple native mussel species. We weren't just looking at threatened and endangered species, which our state and federal agencies pursue, but more of we were pursuing this for water quality. And if you can think of it this way, if you have cleaner water coming into an intake, the less amount of money you have to spend on filtration, the less amount of money you have to spend on chemicals to treat it. So it's a win-win situation for the water department from a source water protection side. We also wanted to reintroduce these mussels into our urban rivers. There are areas, believe it or not, in our urban ecosystems, our urban river systems, that we do have very large and healthy beds of freshwater mussels on the Delaware, some on the Schuylkill, but they're few and far between. There's been a lot of issues historically that have sort of dwindled their populations. Dams being one of the major impetus for obviously for propagation because of fish blockage, gross pollution prior to the Clean Water Act, and so forth. We also wanted to provide a hands-on type of experience concerning mussel ecology at this location. So you get both educational and real scientists doing real type of research and development. And we wanted to establish a location for not only just research for the water department, but for academia, for institutions like Drexel and Penn and so forth, and high schools, and serve as an educational facility for the future production hatching, which I'll get into. So what's involved? And I'm going to show you, if you can follow my arrow, I hope you can. If you can't, if you look at the top picture up top, that is the Glaucidia. Glaucidia is at larval stage. And what happens is that we collect fish, broodstock collection. Each species has a certain host fish. There are some species that have multiple host fish, but there are species that have one fish. For example, the alewife floater relies on alewife or blueback herring when they migrate up in the spring. We collect the larvae in laboratory, inoculate, the process called encystment, and then another process called excisment, and then juvenile rearing. We actually have to rear these things up, grow them to a size where they're not preyed on. So the first process, I guess, is broodstock collection. We actually go out in early March and collect the females. And by doing so, we open them up. This is a non-lethal process. And we look at the mantle or their gills. And then you can see in the lower right-hand corner here is these are the tubes of thousands and thousands of Glaucidia. So we do a manual collection and we identify the females. And then we transport them back to the laboratory and maintain them on a diet of shellfish. And we maintain their temperature or the temperature so they won't broadcast their Glaucidia. At the same time, we're collecting fish too. So we're either collecting alewife, we're collecting largemouth bass. Again, it's species specific. Whatever species of mussels we're trying to propagate, we look at the host fish for them. And we collect them through trap netting or electrofishing. We condition the tanks within the laboratory. As you can see in the lower right-hand corner, all these fish, these are all alewife in there. And then we maintain them for a while. And then comes Laurel Collection. And so we either do it through natural broadcast or syringe method. And syringe method is what we do. Is when we open up the female, you can see the scientist on the bottom, Kurt Chang, actually injecting distilled water into the sacs of the female, and the Glaucidia burst open and they go in there. And then we identify how many of those Glaucidia are alive through a little salt test. So we take a little tiny aliquot, a little tiny amount. We count how many Glaucidia are on that dot. And then we put a little dab of Morton salt, believe it or not, right on there. And if they're alive, they close, they snap shut because they don't like salt water. And then we estimate how many Glaucidia. And in doing so, then we do the actual inoculation, wherein you can see in the top right-hand corner up here, these are American eels. And we basically put a certain volume of Glaucidia in each bucket and agitate the water, agitate the fish for a little bit, up to 5, 10, 15, 20 minutes. And then we keep on checking their gills. And you can see down the lower right-hand corner here, you can see all these little white specks. And those are the Glaucidia attached to the gills. And this actually a gill of the alewife fish. And then that process, once we know that that process has occurred and it's occurred to our liking, our density, if you want to call it, these little, the fish are then placed into this container called a ZHAB, or Animal Habitat Unit. And they basically live out there just relaxing, fed by us. And 0 to 3 days post-inoculation, these Glaucidia start to insist or become, you can see in the lower right-hand corner, almost like a little ring around. And they become part almost of the gill. In doing so, an incredible process takes place. In some species, it's 10 days, like I said. Some species, it's 20, 30, depends on what species. They metamorphose. And they metamorphose into a living, breathing mussel. And you can see right there, these are brand new, newly transformed yellow lamp mussels up here. And then down the bottom, you see newly transformed alewife floaters. And you can see the foot sticking out there. So that's how they get around at that. You know. They also use their foot at that stage to pedial feed, or actually grab algae and bring it into them. So they transform, and then we collect them on the back end of the habitat unit where the fish are, and put them into a thing called a Barnhart system. And the Barnhart system is a very simple 5-gallon bucket with these holes drilled into them with like basically baby mangers, if you want to look at it. And they sit in there, and we are dosing them with a certain amount of algae on a given time every 24 hours. And we're cleaning them out, we're maintaining them. And once they reach around 1 micrometer, usually between 1 and 2 micrometers, we transport them to ponds. And as you can see over here, they're placed in these baskets in the middle with the orange float right here. And they have a sand medium that we put in down there. And after a year to a year and a half, they're fully functional mussels that we can actually tag if we choose so. Tag with a hull print tag, which is just a numbered tag, or radio tag, and then reintroduce into either a wetland, into a stream restoration project, into whatever we're trying to accomplish. So some examples of accomplishments that we did. We started this in 2017. It was our first trial by fire. We successfully propagated 2 known species that are one of our bigger functional dominant species. The eastern liptio, which is 90% of the population in this area is eastern liptio, and alewife floater, which is a huge, a larger type of mussel in the Delaware Basin, usually in the tidal areas because alewives migrate up during the spring like any other Alocine or shad. Again, alewife floaters also use, like I said, alewife, blueback herring, and shad as their host fish to transport upstream. And we also, during 2017, we acquired 15,000 mussel seed and successfully grew them out in our pond. So at the same time we're trying to propagate, we were trying to see, could we grow them out? And we had a very successful crop that year. In 2018, we actually propagated 5 different species at a time. So we see alewife floater, eastern liptio, tidal water mucket. All these species are native to the Delaware drainage. The brood of the females we collected from the Delaware River, in some places in urban areas near the Tacony Palmyra Bridge, down near Schuylkill River, and so forth. In 2019, we had over 10,000 juveniles that we propagated. So this small educational hatchery, it morphed into not only a production type of small production hatchery, but at the same time, thousands of kids coming in, students coming in, and scientists showing them what we're doing and how we're doing it, and the nexus between this and water quality. Some other things is that in 2019, we also found that we could use landlocked alewife instead of trying to hunt for migratory. We don't want to hurt the alewife population because they're already in low numbers. The native alewife populations that are migrating through the gauntlet of the Delaware Basin and out in the ocean. But we found a huge population of landlocked alewife in Blue Marsh Reservoir, which they are actually considered somewhat of a nuisance because there's so many of them. And so we actually could, for the first time, we were propagating using a landlocked species that more or less the Fish and Boat Commission were happy that we were removing. And then we also had successful propagation of mussels using in vitro techniques. And I'm not going to get into that. It's a little complicated, but it's not using the fish. It's using a albumin, or sort of like a blood serum, to propagate without that fish. Now, that sounds like you're playing with God there, but actually it's a really good process for those difficult types of species to successfully propagate. It's something that we're investigating. So what's next in our laboratory? And you can see right here, this is sort of the laboratory setting. What's next is that we want to increase, obviously, optimizing our hatchery. We want to increase production and viability. So meaning we want to increase the number that are propagated, but also that reach that age, that year and a half age where we can reintroduce to projects. Again, refinement of standard operating procedures to make it more slick, make it quicker, faster, more cost effective. Development of new techniques like a toad and in vitro propagation. And we want to expand our educational outreach programs. We have a lot of programs we want to look into. We've been very successful with mussels in schools and mussels in the field, where we actually have given 6 classes, a certain amount of mussels, and they've been monitoring them through the years, even through COVID, and monitoring their growth. And eventually, once they get to a certain size, we'll tag them for them, and they can assist us in restoring a mussel bed. Again, expanding research. The freshwater mussels, again, up until about 10, 15 years ago, the research was pound for pound, if you want to call it, are filtering just as much water as oysters.
Hey, Lance, you froze just for a moment there. You want to back up and
sure.
a sentence or two?
I
I think mussels in the field we were at too, and then yeah.
Yeah, so the Mussels in the Field is another program that we're looking into expanding. We have 6 classes in schools within the city of Philadelphia that we're working with, and they're basically monitoring our mussels, or monitoring their mussels, their baby mussels. And then once they get to a certain size, we're going to tag them for them, and they're going to reintroduce them into one of a stormwater wetland, a stream restoration site, a larger project that we're involved in. We also want to expand our research studies, and also we want to work with Senior Environmental Corps or citizen scientists to promote it when they do have a pond or lake on their property too. These native assemblages or these native mussels are great water filters for them. So we'd like to investigate that too. It's an example of, and then also higher research, looking at different techniques, looking at how to utilize these in our Living Shorelines programs, and then development of a comprehensive strategy for reintroduction. So, you know, you have to have a practical type of strategy when you're reintroducing these. You can't just throw them in the water and say, okay, we did this. You have to know where they can survive and where they can't. Where's the habitat? Where's the habitat optimum for them? Can they survive in this type of stormwater wetland? Is the wetland loaded with pollution too much, or is there too much sedimentation? Is there too much scouring in the stream? So you have to do your homework before you just say, okay, we introduce X amount of mussels and they're removing X amount of pounds of nitrogen per year. So current initiatives, we're trying to quantify ecosystem services in the terms of filtration, like I just said. So looking at pounds of nitrogen or pounds of phosphorus removed per unit or per mussel per given area. Bioextraction is another thing that we're getting interested in, is actually mussel gardening or mussel farming, where you actually put the mussels in at a small size, let them grow really quick, and then take them out. Well, you're actually actively moving X amount of nitrogen and phosphorus in doing that through their mussel and their shell. But then you also utilize those mussels in restoration programs. And then at the same time, you reseed that lake or that pond or that wetland with additional seed. So they start to grow again and again. And it's sort of a maintenance program. One of the bigger things that's happening right now, and this is underneath the partnership with Delaware River, is we're constructing a large production facility hatchery at Bartram's Garden. And that's going to be producing, like I said in my talk, you know, we're producing around 10,000 mussels per year at the demonstration hatchery. Well, they're going to be doing anywhere from 500,000 to 1 million mussels per year. That's incredible. And their goal is not targeting threatened endangered species. Their goal is to target those abundant species that are needed for water quality and habitat improvements. So it's going to be a very unique and a very robust, a very successful hatchery. They've already picked their consultants and they've begun initial designs. They're going through the permitting process, and they hope to actually be producing mussels by 2024. Reintroduction studies in living shorelines programs, which I'll get into a little bit, and stormwater management wetlands, and also riverine restoration programs too. And again, continued research and development. There's always need for additional research in this area. Like I've told you before, the freshwater mussel has been sort of underserved when it comes to research, but in recent years, it's gained incredible notoriety among scientists, watershed managers, and restoration practitioners alike. So again, I don't want to get into too much of the numbers, but this is an example of, these are 4 sites on the Delaware that we surveyed in 2012. And what I'm trying to get here and show you is that based on the amount of mussels that we saw in these 4 sites alone, so if you think of a hectare is equivalent to 2.5 acres. So it is removing, these mussel beds are removing 10 tons of dry total suspended solids per hectare per year. So 10 tons of suspended solids in the water column, 2.5 acres per year. Just think about that over the whole Delaware River if you had multiple healthy freshwater beds. That's an incredible amount of TSS removal. Again, the next thing is the production hatchery I talked to you about, and they're looking to obviously produce a large amount of seed that'll either go out to research institutions, to remedial activities, or when there's an environmental injury occurred through, for example, let's say an oil spill, something like along that line. So they'll actually be selling the seed to different organizations and institutions needing mussel seed to get a return on their investment. the hatchery itself is valued around, or is estimated around $7.9 million, and is funded through Penvest. So they will be growing them out like we do in ponds, and they'll either be doing mussel farming like we're looking into for bioextraction or restoration programs. And then also living shorelines projects. We're actually starting that right now. We've started a demonstration or a study at Bartram's Garden, where we're using oyster bags or oyster shells to create sort of wave break energy dissipation type of structures. And then in doing so, putting mussels behind them and monitoring the success or the relative success of sediment accrual behind the structures and the success or survivorship of the mussels themselves. And these mussels are pit tagged or radio tagged to see where they are. Are they moving upstream, I mean, laterally, downstream, or they stay in their area? And so a living shoreline project is one that, you're using traditional engineering techniques to stabilize or to dissipate wave energy, to stave off erosion from high energy sources. But you're also reintroducing the biology. You're introducing whether it's submerged aquatic vegetation or emergent vegetation or intertidal type of vegetation, wetland or obligate wetland species. In that example too, you're also introducing biology in terms of animals, such as freshwater mussels in our area. So the goal there is obviously to increase habitat diversity in areas that have been scarred or that have lower quality habitat, stabilize the shorelines, provide or increase ecosystem services in the area, and resiliency either towards storm events or to climatic changes that are occurring. So again, I'm going to leave you with that, but this is just these are newly hatched freshwater mussels right there. I don't know if you saw the video. And I wanted to acknowledge part of the ARC technical team that's been helping me and everyone involved to make this program a very huge success. Danielle Krieger, Kurt Chang, Matt Gentry from the Partnership to Estuary, Roger Thomas from the Academy of Natural Sciences, Rick Anthus and Will Whalen from Philadelphia Water Dept, and Bria from Audubon, Pennsylvania, and Shannon Boyle, our Drexel Co-op student. So with that, I leave it to you and any questions.
That's great. Thank you, Lance. That's wonderful. Yeah, we have a question right away from Sandy. Will they be found in creeks like the Wissahickon? I think you alluded to that.
Yes.
And if, yeah, go ahead.
Sandy, that's a great question. The answer is they should be there. That's the reason. We have great historical documentations from a scientist, a guy named Ortman, who did studies in 1911 and 1909 and 1919, I think, on all Pennsylvania and southeastern Pennsylvania. And he found that the richest areas of mussels were actually in our Schuylkill navigation systems and the Wissahickon and so forth. So yeah, to answer your question, yes, they should be there. Have we found any? No, we found it's not invasive, it's not categorized invasive, but it's a non-native corbicula, which is the Asiatic clam. We haven't found any of the invasive species that we're worried about, such as the zebra mussel or the quagga mussel or something like that. But yes, there should be, the eastern elliptio should be there. Certain other species could be present in that watershed, just based on the fisheries population. But again, if you think of the Wissahickon, think about what's at the bottom, dams. We have 2 major dams there. So once the population is scoured out from the top, how can it repopulate itself without the fish flying?
Right. So is ARC looking to assist their replacement into the Wissahickon?
Yeah, that's one of our ultimate goals, is to restore where we can restore. There are certain locations where you can restore these in the Wissahickon, let alone now. There are certain pool areas that the mussels can handle this. There's areas in stream restorations. There's stormwater wetlands. We're actually doing a study at Sailor's Grove where we're actually going to do some mussel gardening or mussel farming there, where we're going to take caged mussels and study them for a year and look at their survivorship over a given year to see if they can handle that pollutant loading or that loading during storm events. So yeah, to answer your questions, Wissahickon is very much on our radar.
That's great. Other than the elimination of a reproductive bottleneck, are there advantages to propagating 5 species of freshwater mussels?
Most definitely. So if you think about just the basic principles or concepts of ecology, any system that has a more complex system or more diverse system is obviously has a greater chance of withstanding perturbation and storm events and pollution events. And if you have more species, you have more species richness, you're going to have that ability to do so. Along that line of thinking too, each one has its own niche in the sense that the species that we're talking about, some of them are generalists in their host fish, some of them are specialists, some of them reproduce in the spring, some reproduce, propagate, some of them disperse in the summer, late summer. So there's all this type of ecological linkages with the fish and so forth. And also creating a mosaic on the bottom of different mussels species is a more complex system and a more stable system.
Right. Barbara asks, what specifically are they being inoculated from? When you inoculate?
So the word inoculation probably is not appropriate. We're basically taking the Glaucidia, the larvae from the female, then actively placing the Glaucidia onto the fish gill instead of it naturally occurring. We're sort of hedging our bets and increasing the likelihood that there's going to be 100, 200, 500 these individuals on the gills of an individual fish.
Right. So I love this notion that mussels can't travel very far. So the evolutionary response is to create Glaucidia, whose job it is to disperse. And we're going to attach to gills. I'm curious about that specific mechanism. Like they could attach anywhere, the skin, the tail, inside the mouth. So is it they're going for the oxygen in the gill perhaps, or?
It's a great question, but what we're finding now is that the gut microbiome, the gut of the mussel is heavily linked to its host fish. So something that we don't necessarily know yet is occurring synergistically. Not synergistically, it's more of commensalism. One's benefiting, and I don't think the other one, the fish, is not benefiting at all. It's not killing the fish by any means, and in anything, too much of a good thing can kill it. So it's this interaction between what the Glaucidia is doing on the gill. There's some sort of exchange, whether it's nutrients, oxygen, some sort of biome that is exchanged with the mussel.
I guess the blood is close to the surface of the skin at the gill, I guess.
Exactly, and when they're attached, they have little teeth, like tiny little teeth on the tip, but hook into the gill. So
yeah, it's so interesting, the biology. That's so fascinating.
It's immunoresponse, basically. Yeah.
So this seems like a labor-intensive project. So where's the funding for this coming from? I'm curious.
So it's not as labor. The capital to start it is extensive. The demonstration hatchery was actually funded through a single grant. I'm not sure the grantee, but it was in the order of $300,000 to start up the educational area. And we subcontracted with Habitat Eats, with Victoria Prizia and Claire Donato and her agency. And they created the living, breathing laboratory. So the other one, the production hatchery, is in the order of, obviously it's 10 times the scale. It's a $7.9 million project. But the actual operation, if you think about stream restoration, the rate on return it's about $156. This is the literature, about $156 per pound of nitrogen for stream restoration. Whereas a mussel, it's $15. So it's an order of magnitude less than if you're doing a stream restoration to sequester that nitrogen in place or to remove it from the system. So it's a very, very beneficial, a nice tool in the toolbox for pollution management.
Philip wants to know, why is Vanskyer Lake being seeded? Is it close to because it's close to a landfill? And for those of us who don't know, where's Vanskyer Lake?
Yeah, Vanskyer Lake is up in Tullytown. That's a great question. We were lucky. We partnered with Waste Management, and they said the general managers there said, yeah, you can investigate this. I mean, they are right next to the Delaware River, and we collect some of our broodstock right at Tully Town, right across from their waste management facility there. And they said, yeah, you can, because they're native, you're not introducing non-native species. You're more than welcome to use this as a broodstock or a rearing hatchery.
That's great. Interesting. That's really great. So you mentioned the American eel, and I'd love the life cycle of the American eel. It's probably one of the fish that no one knows anything about.
Right.
Would you mind walking us through the life cycle of the eel real fast?
Sure.
Because it's just crazy.
Yeah. So opposite of the anadromous or migratory fish that we think of, like the salmon coming out from the saltwater into the freshwater to reproduce, in many cases once and die, like the American shad or the striped bass, right? Or striped bass, any type of anadromous fish. The American eel is actually catadromous, and that's the opposite. So what happens is females go up into these streams, rivers that you see Wissahickon. Pequessing is loaded with American eels, pennypack too. And they basically live out their life cycle until reproduction period, and then they migrate out into the saltwater, and they all aggregate at the Sargasso Sea. And reproduction occurs there, and the female dies, and the young of the year make their way back up to their natal grounds, in many cases right back to the same stream.
Yeah. It's a wild life cycle. But I remember when I worked for the Lower Merion Conservancy, there was a creek there, Mill Creek, that we were doing water quality studies on, and we would do some fish shocking with the Academy of Natural Sciences. And almost every year we'd find an eel, nice 3-foot eel. And this is before the fish ladder. So this thing is like crawling up out of the water around a dam. So it's coming out, going around the dam and coming back in. And I'm going to get to that stream up there. I got to get back to that stream up there.
Eels are amazing. They are they're very adapted to using the interstitial spaces of dams themselves, the faces of the dams, and basically making their way up the dam too, and around the dam and under the dam in any type of way they can get up the river.
Yeah.
They're very, very hardy and very interesting fish.
But there's also like a big mystery about what they do in the Sargasso Sea, because they think that they're there, but they've never found babies. There's some piece of the life cycle they've like never found.
Yeah, I mean, exactly. In the Sargasso Sea, I would defer to our fisheries biologists on that one, but yeah.
It's just funny. I digress, but it's an amazing life cycle. How are the fish themselves doing in our rivers? How are the shad doing, for example?
Yeah, I mean, so again, mixed results. There's the water quality is improving. The Delaware's looking much cleaner since I've started. The Schuylkill too. The fisheries, it depends on what species, it's species specific. So the shad or the Atlantic stock, I think the Atlantic stock itself they're having mixed results. In some years it's high, some years it's low. The Schuylkill is seeing that too. You'll see sometimes at our fish ladder, catch per unit efforts of high or fish passing high and then going low. There's a lot of reasons for that. I mean, there's a lot of subjective reasons for it too. There's, fish passages aren't really made. They were made with the idea of passing salmon instead of American shad. So they engineered them towards salmon instead of American shad. And we've tried to address that. We have a fisheries biologist that's looking into that and monitoring that and see how it's doing. But it's a mixed message. There's a lot. Our species diversity, our species richness in Delaware, we've got over 60 species in the tidal Delaware. Of that, 35 plus are utilizing the fish ladder to go upstream. Now, some of them can use it very readily, gizzard shad and white sucker and other species, whereas other species have a hard time utilizing it. We do have invasive species that are concerning, such as the flathead catfish and the snakehead, because they're pretty well adapted to inhibiting fish passage, meaning that they're big and they prey on a lot.
Right. Okay, good. So we have a few minutes more. If any more questions, please put them in chat. Happy to ask. Let's do one more digression, if you don't mind, because a lot of our people on the program tonight probably have walked by the Manayunk Canal, and the water there doesn't move very much. And there's a bunch of partners, again, working on fixing that situation. We're actually to get water flowing through the Manayunk Canal, which would be a wonderful thing. You want to describe that in sort of broad terms for us?
Yeah, so I mean, it's a multi-million-dollar project to stabilize the wing wall or the area of the upper Manayunk Canal, where the locks are. At the same time, reintroducing water from the Schuylkill to improve water quality within the Manayunk Canal. And you're absolutely right. The velocity of a drop of water from starting from the top, going all the way down to Lock Street and out to the Schuylkill, it takes 4 days of that drop of water to get there. Now, what we're trying to do is get that in 4 to 6 hours. So we're going from 1 to 3 cubic feet per second, which is basically a dribble, to about 170 to 100 CFS. That velocity will then be, right now it's 0.03 feet per second. So again, a very slow moving particle, to about 0.3 to 0.5 CFS, which is much faster. It's not to the point where you can't canoe or kayak through it.
Right.
It's just, it'll flow, it's laminar, it's going to be laminar flow. You're not going to see that filamentous type of nuisance algae in the water anymore in that location. And dissolved oxygen is going to improve, so the fisheries are going to improve in that location. Also, we're opening it up, so fish in the Schuylkill can go into the canal, or fish in the Manayunk Canal can go out to the Schuylkill.
That's great. Is that good for mussels?
Yes, actually, that's one of our study sites. That's actually where my dissertation is going to focus on, is reintroducing mussels there.
Oh, that's so great.
Yeah.
Okay, looking forward to that. Thank you. So, so mussels are our blue-collar bivalves. That's a great concept. Do you see any consciousness about them rising, because they sort of float under the radar screen?
It's actually occurring already through the works of our sister agencies and partnership with Delaware River. Partnership with Delaware River has done great public awareness on this subject, and just the literature. I mean, the amount of literature and the amount of knowledge. I mean, we're finding that people thought that these organisms or these animals didn't filter as well as their marine counterparts, their marine cousins, such as the blue or the blue mussel or the Virginia oyster. And what we're finding is that they filter just as well, even in many cases filter more water. And they don't really take a nap during winter either. They are filtering through winter too. So the ecosystem services that they're finding that these things provide, and what they're finding, which is really interesting, is that because of their movement up and down the sediment, column, and also their microbiology, their waste, their biodeposits, or their pseudothesis, we call it, they're enhancing the microbial loop in that area, which actually increases denitrification, which is really good. So you're taking nitrogen and you're degassing it out of the water in the form of nitrogen gas. So it's a really good process to remove nitrogen from the waste stream, or from the river or lake or wherever you are.
That's so great. Well, good luck with your work. I really appreciate it.
Thank you.
Thank you. And thank you for spending time with us tonight. We really appreciate it. Thank you, Lance.
You're welcome.
Lance Butler is a senior scientist with the Philadelphia Department of Water and Office of Watersheds, working on restoring mussels to our streams and rivers. Thank you so much, Lance.
Thank you.
And everybody, thank you all so much. Next week is our Juneteenth celebration, Black to Nature. And the week after, live in people coming back to the Nature Center on the 24th at 7 o'clock for the opening of Al Mudif. So hopefully we'll see you next week here on Zoom. And then a week after, come to the Nature Center for the opening of the Al Mudif. Thank you also, everybody. Thank you so much. Good night.